It's a pleasure and honor to be giving this seminar. I'm very grateful for this opportunity to be sharing our research and receiving any feedback that you may wish to share. So I thought I would start with a fairly uncontroversial statement, which is that the adult brain is less plastic than the immature brain. It is also fairly established that this is not so much the result of a sclerotization process where the brain loses the ability to change, but rather results from an active stabilization process that I like to visualize as a net that pins the components of the system down in a particular configuration, conferring it with a particular function. There is a lot of research on the cellular and molecular basis of such net. And for example, we know that GABAergic inhibition is probably very important because maturation of the GABAergic inhibition goes along with maturation and stabilization. But I come from discussions from a system level perspective. So I ask, what is it that draws the net and stabilizes the system? And to preview, my hypothesis is that the key stabilizing force is expectations. Because with experience, we come to have very strong expectations for how our sensory world looks like. And this sets the system into a very efficient mode of processing, one that is based on a priori assumptions and that only represents sensory information in a very partial way to the extent that they mismatch those a priori expectations. And although efficient, this mode of processing makes it very hard to learn from new data because they are hardly represented. So this concept brings about the hypothesis that in order to reopen plasticity of the human brain, which is sometimes required, for example, in the treatment of some pathological conditions, what we could do is to play with unpredictability, make the sensory stimuli unpredictable. So this is the general hypothesis that I propose to test. And today I'd like to share some initial steps in this direction. I'm going to be focusing on perhaps the best established case of plasticity, which is ocular dominance plasticity. So we know that during development, the representation of the two eyes in the visual cortex, here represented by these colored boxes, can be reorganized based on sensory stimulation. Because if one eye is deprived of sensory experience, its representation in the cortex shrinks. However, if the eye is reopened while the development is still ongoing, while the cortex is still immature and the net has not been drawn yet, the cortical representation can recover to near normal. However, if the reinstatement of a normal visual experience happens after the closure of the so-called critical period, after the net has been drawn, the cortical representation remains highly anomalous. And what happens is that the cortex does not represent the eye that used to be deprived during development. Even if that eye is now sending perfectly good signals, the cortex fails to represent it, fails to acknowledge it, resulting in a sort of cortical blindness that we know as amblyopia. Now, based on this knowledge, we would predict that manipulating visual experience in adulthood should not produce any cortical reorganization. However, this is not entirely true. There is an effect on ocular dominance, although it is a very counterintuitive one. One of the first demonstrations came from the lab of my mentors, Concetta Morrone and David Byrne in Pisa, where a PhD student at the time, Claudia Lunghi, asked a group of young adults, including ourselves, to wear a monocular patch for just a couple of hours. And she measured their ocular dominance before and after these. In human participants, we often use to index ocular dominance a behavioral procedure known as binocular rivalry. Here, this consists of presenting incompatible images to the two eyes so that they do not fuse in a single coherent percept, but they alternate in perception as shown in this GIF. And what's important is that the proportion of time during which one eye dominates perception is indicative of that eye ocular dominance. So we can use this as a quantitative measure to measure ocular dominance and compare it before and after the monocular deprivation. And this comparison shows that with just 2 hours of monocular deprivation, there is a reliable ocular dominance shift, and it is in favor of the deprived eye. So the opposite of what happens with amblyopia, where ocular dominance shifts in favor of the non-deprived eye. By repeatedly testing binocular rivalry after the patch has been removed, we can also see the decay of the effect and find that the effect is barely visible after about 1 hour from patch removal with this exponential decay, which is again very different from the permanent deficit that is observed in amblyopia. These results have since been replicated by some 100 and more experiments, which we have meta-analyzed to try and get a better sense of the dynamics of the effect. So this graph shows the average effect across multiple studies. The size of the symbol is related to the number of experiments we are averaging across. Most of them were okay, and I'm plotting this, as a function of the duration of monocular deprivation that each study elected to use. And the majority of experiments used about 2 hours or 2 hours and a half, with fewer experiments at shorter and longer durations. The picture that emerges from here is, number one, basically all experiments revealed a shift of ocular dominance in favor of the deprived eye. So this is a very robust and reliable effect. Also, the effect size grows mildly with the duration of this deprivation. And deprivation duration also affects the time that this effect lasts after patch removal. Extrapolating from this curve, we would predict that a much longer deprivation for maybe days or weeks should actually promote an effect that is permanent, so a permanent ocular dominance shift in adult participants. So if this were confirmed by experimental data, it would actually counter the argument that there is no ocular dominance plasticity past the end of the critical period. And I'll come back to this at the end of my talk. Okay, the effect of short-term monocular deprivation has not just been measured or indexed with behavior. It has also been measured with physiological techniques like EEG or fMRI. And for example, here is a map of bold fMRI bold responses elicited by a monocular full field stimulation. These are data we collected with the driving force of Jan Kurjawski and the collaboration with the Imago 7 Center in Pisa. So from this map of activity, we can extract responses to the stimuli in a predefined anatomical region, for example, the primary visual cortex V1. And we can check the amplitude of responses to the stimuli in each eye, dominant and undominant, before and after the application of the monocular patch for 2 hours. And what we see is that the response to the deprived eye before deprivation is fairly similar to the response in the non-deprived eye or non-dominant eye, and that's consistent with the fact that our participants were normally sighted, non-amblyopic. However, after the 2-hour deprivation, there is a clear enhancement of the deprived eye representation compared to the non-deprived eye representation, which is consistent with the shift of ocular dominance in favor of the deprived eye that we've seen psychophysically. Now, because these data were acquired at 70 ultra-high field with ultra-high field fMRI, we can also quantify responses in these much smaller nuclei that compose the visual thalamus, and specifically two regions, the lateral geniculate nucleus that provides the main feed-forward input to the primary visual cortex V1, and its neighbor, the pulvinar here, the ventral section of the pulvinar, which has a completely different physiological role and primarily receives information from the cortex and sends it back, forming the so-called cortico-pulvinocortical loops. Okay, so if we quantify responses in these regions, we see that the responses in the lateral geniculate nucleus are perfectly unaffected by the monocular deprivation, indicating that this simple protocol does not affect retinal responses and it does not affect activity in the main bottom-up input to the primary visual cortex. However, the adjacent pulvinar shows a modulation which is very similar to the one seen in the primary visual cortex, potentially implicating this structure in the generation of the monocular deprivation effect. And this structure is going to come up again very soon in my talk. Okay, so we have, summarizing these results, we have some 10 years of research showing that patching one eye for a few hours in adult humans leads to a shift of ocular dominance, a boost of the response in that eye, which is seen both in behavior and with physiological techniques. And this effect has been generally understood as a homeostatic response. So again, enhancement in response to the decrease of sensory stimulation during the application of the patch. Just like the cortex was pumping up the volume in order to try and listen to the missing signals from one eye. However, and here comes the plot twist, we have come to realize that there is also a very different interpretation possible for these results based on predictive coding, an emerging theoretical framework. So this popular framework essentially assumes that responses in the sensory cortex are generally suppressed unless they mismatch their predictions. Or to state it differently, the idea is that sensory signals are never represented in full, completely, but only to the extent that they mismatch our a priori expectations. Now, a patched eye is an unpredictable eye in the sense that it fails to signal events that I know full well that they are happening, for example, because I'm making them happen with my actions. So this unpredictability could in principle explain the homeostatic enhancement and the ocular dominance shift. If this, so this is my hypothesis, and then the question becomes, how do we test this hypothesis? Well, if this explanation is correct, then it follows that this ocular dominance shift could be generated without changing the strength of the bottom-up input, without depriving the eye, but selectively changing the predictability of the signals coming from one eye. And in 2 experiments, we have collected a proof of concept of these. So we used altered reality, an altered reality setup, to manipulate signals in the 2 eyes without changing their quality. In this system, there is a set of cameras that collect an image of the participant's field of view and place it to the participant's eye eyes inside the oculus. Now there's two cameras connected, each connected to one eye, and one eye is seeing the images in real time, whereas the other is seeing the images delayed by a very long time, 1/3 of a second. And this is more or less what the participant would see. The same image of himself or herself making actions in their visual field. These actions, the task that a subject is asked to perform, is a fairly simple visual-motor coordination task. It's like playing Jenga. They are building towers and they're trying not to make them fall. And this requires eye-to-hand coordination because if the eyes do not give correct information about the position of the hand is likely to be intercepting one of the towers and making them fall. So in this sense, the delayed eye is useless for the task. In fact, it's harmful for the performance of the task because it is signaling the hands at an incorrect position, the position where they were 1/3 of a second ago. So participants played this game with this delay, monocular delay, for 1 hour. And before and after this, we measured binocular rivalry, so ocular dominance through binocular rivalry. And what we see is that there is a shift of ocular dominance, which is in the same direction of the shift we typically observe after monocular deprivation. But remember, here we're not depriving anything. We are not changing the quality of information in either eye. This effect has been demonstrated in a first data set of 20-something participants, and we have recently replicated it in another set of 20-something participants. So this is another reliable and robust effect, although perhaps counterintuitive. One important thing to notice is that the monocular delay alone was not sufficient to generate the effect because there was no ocular dominance shift when participants were exposed to the same monocular delay for the same amount of time, 1 hour, but they did not play actively, make actions in their visual field. They were either watching and listening to the experimenter performing the same task, or they were seeing a replayed version of the video we collected during the first experiment where they were playing with the building blocks, with the wooden blocks, with the monocular delay. So the exact identical visual stimulus in the delayed eye and the non-delayed eye was presented in these two conditions to the same participants, but only in the condition where the participant was actively engaged in the visuomotor coordination task did we see a shift, a reliable shift of ocular dominance, and there was no shift of ocular dominance in the passive viewing condition. So why is this happening? This is happening because when I'm watching passively this video, again, I'll play it from before, I have no way of telling which eye is right. I see that there is a difference between the two eyes, but one could be offset, either one could be offset. Making actions, on the other hand, generates very strong and precise expectations for where and when visual changes should happen. And so it unequivocally tags one eye as predictable and the other eye as mismatched. I'll take this opportunity to point out that the manipulation imposed by this monocular delay is really very subtle in the sense that the images in the two eyes are coherent to a very large extent, and monocular delay only introduces mismatches in those portions of the visual field where a change occurs, whereas all the static portions of the visual field are perfectly coherent between the eyes. In case you're wondering, eye movements cannot be contributing to this mismatch because as long as the images out there are static, the monocular delay does not have any effect. It's as though it didn't exist. So moving your eyes across a static scene will not contribute to the mismatches observed. And it will only be the changes produced by the participants' own actions that produce mismatches between the eyes. Okay, so we're not changing the quality of the information. The two eyes are getting the same information, just delayed. And we are producing mismatches that only affect 20% of the pixels. This is how we estimated it. So how strong can this effect be compared to the classic monocular deprivation effect? To allow for a fair comparison, we added a third condition where we again replayed the same participants, the same movie that we had collected in the first condition when they were actively playing with the building blocks. But this time we replaced the delayed image with a homogeneous gray screen, effectively removing visual information from that eye. So this gave us a measure of the monocular deprivation effect for the same participants and the same amount of time, again 1 hour, revealing the expected shift of ocular dominance in favor of this time, deprived eye. And we, to our surprise, found that the size of this effect was essentially the same as the effect of monocular delay while actively engaged in the task, and that the two effects, the effect of monocular delay in active vision and the effect of monocular deprivation, was very well correlated across participants. So to me, this suggests that the unpredictability of the deprived or the delayed eye could lay at the basis of both effects, potentially accounting for this correlation. Okay. To account for this new sort of effect, monocular delay, the homeostatic model is not a good model. Let me show you how this would work. So in the homeostatic plasticity model, there is a reduction of input in one eye during the application of the patch. The cortex responds to this reduced input by increasing the gain, the response gain, and bringing back responses to about the same level for the two eyes, which is homeostasis. But then after patch removal, this enhanced gain produces transiently increased responses to the now recovered normal input, bottom-up input. In the monocular delay experiment, however, there was no change in the bottom-up input. So it is very difficult to explain the enhanced response to the delayed eye. And because the bottom-up input wasn't changing, what I expect is that the top-down modulation might be changing. My hypothesis is that because the delayed eye was systematically mismatched with the expectations generated by actions generated by the multisensory context that goes with them, the impact of these predictions gradually reduced, and this resulted in a boosted response, given that we know that predictions tend to suppress responses in sensory areas. So it's interesting that in this second scenario, we are assuming that the origin of the effect is in the top-down prediction. So it's somewhere high level linked with action planning and organization. And nevertheless, we have to come back down to V1 to generate the ocular dominance shift, simply because V1 is one of the few places where the two eyes are separately represented, and so one can be boosted without an effect or in the opposite direction than the other. So at this point, you might be wondering how likely it is that predictions and multisensory signals feed all the way back down in V1. And I would say that there is actually fairly strong evidence for this, for the observation that V1 responses in primary visual cortex V1 are shaped by the multisensory context and they are reduced for predictable stimuli. The origin, however, of these predictive signals and the circuit that makes this possible is complex and far from understood. And in particular, it is possible that these modulatory signals come from other areas directly, so through cortico-cortical connections, but it is also possible that part of these signals flow through subcortical regions. And the pulvinar, here PL, is particularly well positioned for supporting this kind of communication because it receives from a large number of areas and it projects back to the cortex, including the visual cortex. Also, we know from basic physiology that pulvinar to cortex inputs are typically modulatory inputs. They do not generate de novo responses, but they typically modulate pre-existing responses, and they often inhibit responses generated by bottom-up input, which is exactly what we would expect for a predictive signal. So putting these basic physiology findings together, we generated the hypothesis that short-term monocular deprivation and other monocular manipulations like delay may be affecting V1 responses and ocular dominance by interfering with its pulvinar connectivity. To test this circuit model, we went back to our 70 fMRI collaborators, and this time the driving force was PhD student at the time, postdoc now Miriam Acquafredda. And this time we recorded bold signals before and after monocular deprivation, but without any sensory stimulus. We asked our participants to lay in the scanner in resting state with their eyes closed. Now, if monocular deprivation selectively affected the gain of visual responses in V1, we would not expect any changes in the bold signals when there is no visual stimulus and we are not recording any visual responses. However, what we found was a very obvious change in the resting state pattern of connectivity between the occipital cortex and specifically the pulvinar. So this is a representation of connectivity between the cortex and the pulvinar. I'd like to show this to you in a more detailed way in the next slide. So here we are looking at the connectivity between the pulvinar and the cortical mantle, average across many participants and across the two hemispheres. We see that the connectivity is very strong in the occipital lobe, as well as in other primary sensory areas, including the temporal and parietal areas, which is entirely consistent with the physiology of the pulvinar as a multisensory information hub. This other set of images show the difference in connectivity before versus after the monocular deprivation. And blue means that connectivity was radically and significantly reduced after the monocular deprivation, and that happened almost selectively within the occipital lobe and its medial wall, in fact. This is a quantification of the effect in the anatomically predefined primary visual cortex region of interest, showing a very significant change of connectivity. And of course, sitting next to the pulvinar is another interesting region of interest to look at, which is the lateral geniculate nucleus. So we quantified the connectivity of LGN in the same way as we did for the pulvinar. And before deprivation, we see that the pattern of connectivity is much more focused to the primary visual cortex, which is entirely consistent with the physiology of LGN. However, there was no change in connectivity after the monocular deprivation. which is again consistent with the concept that monocular deprivation does not affect the bottom-up input to the primary visual cortex. We extended these results with a second set of acquisitions, which we collected at a relatively fast pace, only 1 second, 1 volume per second. And that's a relatively fast frequency for fMRI. Because we enhanced the temporal resolution, we had to decrease our field of view, and we could only collect signals from this small slab across the brain, which nonetheless included the majority of the primary visual cortex and the 2 key regions that I've been discussing, the pulvinar and the lateral geniculate nucleus. Here we are looking at a map of the connectivity of V1 with the thalamus, which is sort of the opposite perspective from the results I've been showing before, where we were looking at the connectivity of the thalamus with the cortex. Here is V1 with the thalamus. Before deprivation, we see 2 clear hotspots of connectivity, of V1 connectivity, one in the LGN, again, entirely consistent with physiology, and another one with the pulvinar, as expected, given its role in mediating corticopulvinar connectivity. What's interesting is that after deprivation, the hotspot in the LGN was essentially the same, but there was a strong decrease of connectivity in the pulvinar, which is entirely consistent with the data set that I showed you a minute ago. Based on these data and taking advantage of their relatively high resolution, we could model the ball time courses with a statistical model known as the dynamic causal modeling, which allows us to test or to gauge an indirect index of the directionality of communication. So we've seen that the communication between V1 and the pulvinar decreases after monocular deprivation. And we can now ask the question, is it because V1 stops talking to the pulvinar, or is it the other way around? So we modeled the time courses with this simple 3-region model, essentially modeling the communication only between V1, LGN, and pulvinar. And we estimated separately the strength of connections going from each of the thalamic regions to the cortex and vice versa. We then tested the strength of each of these directional connections after monocular deprivation, which is shown here. And we found that the only connection that showed a reliable change, a reliable decrease, was the connection from the pulvinar to the primary visual cortex V1, which went radically down after monocular deprivation, whereas all the other connections, V1 to the thalamus or LGN to V1, remained unchanged. Okay, so this is evidence that that manipulating visual experience for just a short period of time, these 2 hours, was able to profoundly affect the resting state connectivity of the brain, the visual brain in particular. We can, based on this data, on the data that have been presented so far, we have developed a system-level model that combines evidence from the literature with our experimental results. So we know from the literature that the pulvinar collects modulatory signals and predictions from multiple areas in the brain and conveys them to the visual cortex. We also know that these predictive signals have an inhibitory effect. They suppress responses to matching sense bottom-up inputs. And we hypothesize that this results in a stabilized sensory circuit, in a stabilized visual cortex, like a net drawn over the cortex. Now, with a short-term monocular deprivation or monocular delay or other monocular manipulations, we hypothesize that the connection from the pulvinar to the cortex is reduced, it's interrupted, as supported by the fMRI results that I just showed. And this results at least two important consequences. Number one, the reduced inhibition can account for the boosted responses in the visual cortex and the shifting ocular dominance that we started with. However, there could also be more long-term consequences because, as I argued at the beginning, working in a predictive mode is efficient, but it prevents the cortex from representing information, and it therefore prevents the cortex from learning from new information. So breaking free from this predictive mode, from these predictive signals, could endow the visual cortex with a new window of plasticity. And we can test this hypothesis by going back to the amblyopia case that I presented at the beginning. Let me remind you that amblyopia is a case where in adults we see abnormal vision in one eye, meaning that there is less ability to read from one eye, for example, which is linked to a history of abnormal vision in early development, but it is not caused by a current abnormality in the optics or the retina of the eye. So the idea is that an impairment, like for example a cataract, which persisted through the development and it was not treated within that critical window, results in an impairment of the cortical representation of the eye, even if the treatment eventually occurs, if the treatment occurs after the net has been drawn and the cortex is stabilized. So then our question becomes, how can we make the cortex listen to this recovered eye and reevaluate the ability of this treated eye to send the perfectly good information that it is generating? We know from past research that the typical approach to amblyopia, which is the patching of the good eye, is not effective in adult amblyops. It works fairly well during the development, but it doesn't in adults. However, recent evidence indicates that a counterintuitive treatment can actually permit a partial recovery of acuity in the amblyopic eye. And that's patching the amblyopic eye, which is actually very, very convenient in a sense, because this eye is the eye that the patients are relying the least about. And so compliance with this kind of patching is certainly better, higher, or easier to reach than patching the good eye that the patient is relying upon for any of their everyday activity. So the question is whether repeatedly patching the amblyopic eye can lead to improving its acuity. And we have previous evidence in collaboration with Dr. Alessandro Sale at the CNR, Institute of Neuroscience of the CNR. We have evidence from previous papers that Dr. Sale participated in showing that this counterintuitive approach can partially rescue acuity in the amblyopic eye. And we, in collaboration with him, we are implementing a new study testing the impact of amblyopic eye patching performed repeatedly over a 1-month period. This is the outline of the protocol. So the patients wear a patch for multiple times during the month, and every time they wear a patch, they are performing their exercising on a stationary bike. And this is because previous evidence suggested that this physical exercise have the capability of enhancing the plasticity effect. So before and after this 1 month training, we measured behavioral responses, binocular rivalry and acuity, as well as fMRI responses in 10 patients, all of them anisotropic amblyopes. And today I will not be sharing the fMRI analysis, but will focus on the behavioral data. So we'll start from the binocular rivalry, which shows that the initial complete dominance of the fellow eye was gradually reduced after the training. Meaning that the fellow eye was no longer completely dominating perception. The amblyopic eye, to be fair, was not able to dominate rivalry, but it was at least able to enter the so-called mixed percepts, which are times when a combination of the images in the two eyes, often like patches of the two stimuli, enter conscious perception. We then looked at the acuity, and we found that the acuity measured before and after the training in this patient was consistently significantly better, and it was improved by about 1 line, which is not a large improvement, but it is a significant one. And also, it indicates, it suggests that the improvement is long-term, because some of the study is still ongoing, but some of these patients were able to complete the protocol more than 6 months ago, and we have another measurement of their acuity 6 months after the end of all these training protocol, and these patients are still showing this improved acuity. So this is indicative of a long term of a stable acuity enhancement. Of course, the way I interpret these findings is assuming that patching the amblyopic eye made that eye unpredictable, and this could be the key to enhancing its cortical representation. Why? Because this eye became became mismatched with the top-down predictions. Now, if this is the explanation, however, it is obvious that monocular patching is a very suboptimal approach. Because if the aim is to let the cortex know that the amblyopic eye is sending perfectly good signals and it should be listening to it as it does for the other eye, reducing the quality of the signal through patching is certainly not the best way. And a much more effective strategy would be to make that the amblyopic eye unpredictable without depriving it, for example, through monocular delay, which is, of course, the next step we would like to implement. But at this point, it's definitely time to summarize and conclude. Today, I presented our attempt to interpreting sensory plasticity from the point of view of predictive coding. And although this is certainly unconventional, the underlying idea is really quite simple, that strong a priori expectations discourage learning from new data. We see this in science, we see this every day. And my proposal is to apply this concept to early sensory processing, which could enhance our understanding of plasticity and potentially our ability to harness it for therapeutical purposes. In the set of studies that I presented, we've been focusing on ocular dominance plasticity. Why? Because we think it's a good model of brain plasticity because we have reliable and precise tools to measure it, and we can test it both in the short term in the normally sighted participants and in the long term in the amblyopic patients, adult amblyopes. Based on these findings, we have been developing a system-level models for understanding plasticity and predictability. And the idea here is that there is a hub, possibly in the pulvinar, that collects predictive signals and relays them to the sensory cortex. inhibiting responses to predictable stimuli, and thereby drawing a stabilizing net over the cortex. Repeatedly mismatching these predictions, however, can lead to reducing the impact of this predictive processing on cortical processing, making it less predictive, more data-driven, and therefore more plastic and open to learning. This suggests that sensory unpredictability, as achieved by monocular deprivation and other monocular manipulations, could open a new window of plasticity during adult life, past the closure of the so-called critical periods. There are, of course, multiple other questions. Maybe I'll just mention a couple that are very relevant to me. Number one is, if predictions are so important, which is their source? Where are they coming from? And the Oculus experiment I presented at the beginning is giving us a hint that actions are really very important. And this, of course, fits with the classical physiological literature on corollary discharges, those signals that accompany actions. And what do? They suppress sensory responses that are consistent with the action, that are generated with the action. So in this sense, they could be a very strong and predictive signal that informs the sensory cortices. Thinking along these lines could help us understand why visual plasticity appears to depend on physical activity in some very mysterious ways, like, for example, in the in the study on amblyopes that I mentioned before. And it might be that it's not about the muscular exercise, but it is rather about the corollary discharge signals that accompany actions. And those actions could easily be playing Jenga or building wooden blocks, as in the Oculus experiment that I mentioned. And then the other question is, what part of the brain is detecting the mismatch between predictions and sensory data? And I believe that there is room for at least another component in this circuit that signals prediction errors. The physiology literature is suggesting, recent data suggests that diffuse monominergic systems could be very important in the generation of such global error signal. And in humans, we could be ideally positioned to monitor those signals with neuroimaging, of course, as well as with pupillometry. So although this is certainly a simplistic model, my hope is that it is helping us to ask useful questions about plasticity and testing its relation with this increasingly popular framework of predictive coding. So I would like to end by thanking my collaborators, especially the many scientists who contributed to this work. In bold at the top, the students and postdocs who were the driving force in the experiments I presented. Several key collaborators from Imago 7, from the CNR, EET, and also my mentors, Professor Morrone and Burr. Thank you all for your attention. Here are the funding bodies that made this research possible. Thank you.